separating bioactive ingredient would have potential for functional recovery. To look for a new ingredient, botanical ingredient, or pure natural compounds, we have to organize different steps for our study. The first is ethnobotanical studies. We anime our network since 30 years with more than 50 members from 30 different countries. In this ethnobotanical study, ethnobotany is referred to the study of a plant that's human use for various purposes, whether medicinal, nutritional, environmental, artisanal, or otherwise. It therefore combines botanical sciences with human sciences. Its scope is international and extends to all historical periods and geographical regions. The approach of identifying plants of diverse interest will therefore involve, as a first step, an ethnobotanical study to list the plant, their use, and their potential benefit. In this study, ethnobotanical research and plant use and traditional use must therefore cover several aspects. Logistical equipment, data collection, this is a science. This is not human research. This is not literature research. This is very science tools, very science approach. Specify the source of information, identify the reasons for traditional use of plant, food, health, dyeing, magic, environment, perfume, and specify the use of plant. Some plant may have different traditional use, depending on the region. Identify and describe the objective of use, favor, pain, card. This part is very important because we can to avoid confusion over symptom with this description is exact. After of this part ethnobotanical study, we have botanical study, pure botanical study with the identification of plant, floristic survey, common name and the scientific name, plant authentication, purely botanical approaches, macroscopic identification, and microscopic identification. This step led to development of quality certificate and traceability and authentication for plant. Because if you use Boswellia, you can't use Boswellia in place another. We have to be sure that this is Sakura or Serrata or other. That's why we need this traceability and authentication. And we have to study method of cultivation, preparation, and use. We have for this preparation, aqueous extraction, decoction, maceration, infusion, percolation. All methods must be exactly described. It's important to know age of use plant because when a plant is young, shoot, or adult, results is not same. Pharmacological, phytochemical results might be different. We have described part of the plant because in the plant we have root, leaf, flower, trunk, resin. In summary, we have to describe plant identity, part of the plant used, preparation method, traditional properties. And this summary can to give some opportunity to traditional use to laboratory experiment. In the laboratory experiment, we start by extraction, characterization, identification, and purification. We can to use some tools for this purification. For example, CO2 supercritic, it's very adapted for green extraction. As you know, in natural plant, natural biomolecules might be primary and secondary metabolite. Primary metabolite is glycid, protein, is some principal metabolite. And the secondary metabolite, this is our interest. This is a part of to look for a new biomolecules. And the water, which present maximum of the plant. In the secondary metabolite, we know some of this secondary metabolite. In the health and medicinal plant, you have alkaloid, terpene, you have coumarin, glycoside, you have tannin, you have saponin, you have very lot of these biomolecules. I think that 60% of pure drug is extracted from plant. You have in food and the nutraceutical use, this part is new, is progress with the important level. When we want to extract biomolecules, new biomolecules, we have to use some solvent, organic solvent. This extraction depends of polarity or apolarity of this biomolecule, this ingredient. When we use some solvent, as you can to know, organic solvent, we can to extract pure or substantial compounds. But when you use this solvent, we have extraction by solid, like liquid, solid, gas, solid, and with numerous process, decantation, centrifugation, crystallization. But problem with this solvent is not recyclable, formation of toxic byproduct. This why when we use a solvent, we have to be very careful with toxicological effect of this solvent and the all part of the body. In laser reading to quantify a part of each granulometric family. We know identity card of plant, origin and cultivation, medical application, commercial product, scientific research. And we will process by 3 levels. First, this process of separation to prepare powder, fraction, granulometric fraction of the resin. And in the second level, we will study on this fraction all comparative characterization and quantification of chemotype of each granulometric family. In final level, we study toxicological and pharmacological evaluation. This is for to establish a link and relation between process, chemical profiles, chromatographical profiles, and biological profiles to know what fraction will be more important for to use in the commercial development. In the first level, we had make application for our process with the patent, and we obtain 6 family fraction with the reading by laser, and we have this yellow curve for Boswellia, and you can see by this distribution that we have 6 family of 100, between 100 200 micrometers is more important by Y distribution. We establish this time because it will be used for chemical and pharmacological toxicological study. When we will study phytochemical analysis, we will use this fraction, and we will use a marker, tracer of Boswellia. One of more Boswellic acid, one isomer. What name is AKBA. This is one of more isomer acid Boswellic. Why use this? Because we need calibration of our device, chromatographical device. You see here some profile, chromatographical profile of powder without giving, and 50 micrometers family, 100 and 1 to 203 micrometers family. And we compare between this fraction, and we compare with the hydroalcoholic extract for Serrata, not Boswellia, just to know if the process, hydroalcoholic process, is more important than green process. You see here, between 1 and 200 micrometers, we obtain more a part of IKBI of this family. And we compare with the Boswellia serrata, we have important quantity of this tracer in this family, like you see here. And what do now? We will release some toxicological and pharmacological study on the all granulometric family to know where is the more activity. the more bioactivity and the low toxicity to compare it. Now, I need Dr. Lambert to present this level for study. And I can to repent on your question after, if you want. Miss, thank you, Mr. President. I come back for
Dr. Claude Lambert. Yeah, sorry, I didn't see you. Yeah, welcome, sir. Now we have the presentation by Dr. Claude Lambert.
Thank you very much. Thank you for the organizer to invite me and for the organization of this meeting. So you have been introduced about the issue we are addressing, and the idea is that we are going to work on frankincense in immunological part. So there is two main immunological system in the body. One is the innate system, which is not non-specific. Which is non-specific. This managed with all type of particles that you can find in the nature and you are exposed to, and these cells are able to catch the particles and to phagocyte it and to destroy this particle by using many free radicals. These particles can be bacteria, of course, and this is the main way to destroy bacteria, but it can be any type of microparticles. The ingestion of microparticles induces the production of inflammation. The other part is the cognate immunity, which is very highly specific with a good memory on the production, on the defense, and it is well focused on virus, on cancer, on allergy, etc. This include the different type of cells, and we are mainly focusing on T cells to be short. And there is two main population of T cells, the CD4, CD8. CD4 cells are more regulators, CD8 cells are more effectors to deal with the risk. And so they produce a specific response. T cells also, sorry, I'm at the end of the talk already. The T cells are participating to the inflammation also. I suppose, sorry for this. So the main issue to work with resin is that resin are solid, and we cannot use it ex vivo to stimulate the cells, and they are highly hydrophobic. And you have heard about the use of solvents we want to avoid. So we started by using the powder, and we have made some fine powder from the block at the initial step. Then we have dissolved the powder in a solvent, and we centrifuge the solvent, the dissolution, and then we can make the separation between the extract and the rest of the resin which is not dissolved. So we have tested different solvents, and as you have heard, we decided to use DMSO, dimethyl sulfoxide, which is compatible, biocompatible with by using with cells, which is not the case with ethanol or methanol or other buffer. And the second step, as you have heard, we are also testing the different size of the powder that from the extract to see if there is a difference. So we have used, as a readout, we have used flow cytometry. For flow cytometry, we can first we expose the cells, then we can analyze the cells in the machine, and the machine will give you a result of each cells. Each dot you can see on the dot plot, it represents one cell. And then we can select the cells, and we can identify the cells, and we can read out the effect of the product on the cell. So the first thing we have observed is that the cell survival is very dependent on the solvent we have used. You can see in this picture that we can identify and we can count the cells at the end of the experiment, and then we can measure, we can identify the cells a bit further, and we can measure the activation of the cell by using DMSO. You can see that we have a strong destruction of the cells. We don't see any more of the cells, and then it's very difficult to identify the cells and to do the measurement of the cells. If we look in this picture, we have counted the cells, and you can see that using 1% of DMSO, we have a strong destruction of the cells, and we do not have a destruction if we use only 0.1% of DMSO. This is what we have used later. Then we have analyzed the production of free radicals. Free radicals are very active in the body to destroy the bacteria, but also in inflammation. And we could induce the production of free radicals. Free radicals can be triggered by bacteria, by particles, and this will induce the enzymatic production of the different free radicals. And if we use a reagent that is dihydrorhodamine, then the dihydrorhodamine will be oxidized, and it will become fluorescent, and it is easy to detect on the flow cytometry. So we started with whole blood. You will see it's a very easy experiment. We put the reagent, the DHR. We put the stimulant, which is E. coli, but it can be any type of particles. The E. coli are very active. And then we put Boswellia powder extract to test it. We incubate only 15 minutes at 37 degrees, and then we can prepare it to pass on the machine, and we directly have the readout. So you can see that we are working directly on the whole blood, and we can focus on the granulocytes, which are the most active phagocyte in the whole blood. And if we measure the fluorescence produced by the granulocyte, if the granulocytes are not activated, you see that the quantity of fluorescence of DHR is very low. Okay, if we use DMSO, we do not have a difference in the production of DMSO, but if we stimulate the cells, then we still have the same number of granulocytes from the same volume of blood, but we see that we have a strong expression of fluorescence, which is triggered by the presence of E. coli. So the first experiment we have seen is that first, we don't see any difference in the number of cells. This means that the DMSO and Boswellia are not toxic for the cells. And this is on resting cells. And after stimulation, we see that there is no significant difference in the number of cells. So we can consider that it's not toxic. And we have checked the quantity of fluorescence produced by the different conditions. In the non-stimulated cells, we don't see any difference between the samples and the samples with DMSO. We don't see any difference in the production of free radicals, which is very low because of the cells are not stimulated. If we stimulate the cells, the scale is much, much higher. And we can see that still we have some difference between the stimulated cells and the stimulated cells in presence of DMSO. And you can see that we have some difference with DMSO produced at 1%, but you know that it's a bit toxic. But we still have some difference by using the DMSO at 0.1%. So if we compare the production of free radicals, we can see that there is some difference between cells that are non-stimulated, cells with DMSO, and cells with different concentration of Boswellia sacra. We can see that we have a sort of protection of the cells by using Boswellia sacra. If we activate the cells, we can see that the number of cells which are producing free radicals is a bit lower because of the toxicity of DMSO and the stimulation. If we use stimulation, DMSO stimulation, and the different level of Boswellia sacra, you can see that we have a bit of recovering of the cells. And we also have tested the production of free radicals by using different fraction of granulometry. You see this is the full production, full population of Boswellia sacra, and this is Boswellia sacra with granulometry below 50 microns and with 100 micron, 200 micron, and 300 micron. You see that we have some recovering of activity of the cells by adding some Boswellia sacra, depending on the dose we are using. You can see that if we put too much Boswellia sacra, we may have some adverse effect. We have also compared it on resting cells, and the resting cells are not much influenced by the presence of Boswellia sacra with a different granulometry. Then we move to the cognate immunity, and we have analyzed two different populations, the CD4, which are regulatory cells, and CD8, which are the effector cells. In this case, we cannot use directly whole blood, so we have to separate the cells from the red blood cells. So there is a cell separation process, and so we washed away the plasma and other cells. This takes a few hours, and then we can incubate the cells. We can add a stimulant of the cells, which is an antibody, which is quite physiological, and we add or not Boswellia in DMSO 0.1%. In this case, we have to incubate the cells for overnight, one or 2 days. So we have focused on 18 hours, one night, at 37 degrees in conditions for cell culture. Then we can label the cells, and we can analyze it directly on flow cytometry. So here is the readout. We do not have granulocyte. We have only monocyte and lymphocyte from the cells. We can separate the lymphocyte T cells, and from the T cells, we can separate both CD4 or CD8 T cells, and then we can measure the readout of the different subpopulations by measuring the activation of these different cells. It's very precise, it's very rapid. We can analyze more than 300 to 1,000 cells per second, and we can have very precise quantitative results. So the first thing we have checked is that we have checked the number of cells after the stimulation of 18 hours in presence of DMSO. If we culture the cells alone, we have some quantity of the cells. If we culture the same quantity of the cells with DMSO, you see that we have a reduction of survival of the cells. And if we stimulate the cells, as compared to this, we still also have some reduction of the number of cells, so we have some killing of the cells. And if we have the DMSO and the stimulation, we still have more loss of the cells. And if we do the culture with cells DMSO and Boswellia sacra at different dosage, you can see that we have some recovering of the toxicity of the cells due to DMSO. If we do it on activated cells, you can see that we have a loss of cells on activation with DMSO. If we add some Boswellia sacra, we have a good recovering of the cells, except if we have a very strong concentration of Boswellia. If we look at the activation of the two populations I told you about, the CD4 T cells and CD8 T cells, you can see that we have an effect on the stimulation. This is the level of activation of the cells. We always have a difference between CD4 and CD8, but we have a good activation even in presence of DMSO. And if we add Boswellia sacra at different concentration, we are not a very different, very big change as comparing to Boswellia alone, to DMSO alone. If we compare the different fraction of granulometry according to the size of the powder, we can see that you have three concentrations of Boswellia, 0.1, 1, and 10 microgram per microliter. And if we have the basic powder, we can compare it with 50 micron, 100 micron, 200 micron, and 300 micron size of a powder that we have separated. We can see that there is some effect at 100 microgram micrometer powder, and we did it blindly. We did not know about the analysis of a different fraction. If we work on resting cells, we can see that we also have some effect, difference in the effect according to the granulometry of the fractions. So in conclusion, we can conclude that it's very important to manage the concentration of DMSO because you have some effect, some toxic effect on the cells, and you lose part of the cells, and especially if you cultivate your cells for several hours. And we also, but if we use a low concentration of DMSO, the effect is not so strong. We lose the cells, but we lose also some activity of the cells. The second part is, the second conclusion is that Boswellia sacra partly protect the cells from toxicity. The next point is that we also have a protection under activation on DHR production, of free radicals production, and also on the specific immune response. And we also could conclude that we have difference in the activity of Boswellia sacra as according to the different granulometry of the samples we have used. Still, we have to improve the experiment to have more samples we can use, because you have seen that we have many tested 5 or 6 samples. In this condition, we have done a lot of preliminary work, and we have to to go further to understand the mechanism of this toxicity and the protection also. And so the work is still under the process, and I thank you very much for your attention. Just a
discussion in the final
Okay.
of two presentations.
Any question?
Very exciting, and I just have a couple of questions. So, do you have a plan to test the cytotoxicity of bacterial toxins and other toxins other than that CMSO, against, you know, the protective effect of frankincense against other toxins?
Thank you. It's a good question. Yes, we have to test the different stimulus. It can be LPS, endotoxin. Can be also particles of pollution, something like that.
My other question is about the particle size. So, can you explain, as I understood, the smaller particle size, the 100 microliter, has a better effect than the higher particle size?
It's not exact. The extraction from the smaller particles is better, so we have different effects. Okay, but it's a process before we extract in the MSO.
Is it related to the extraction process, or
Yes.
the particle size, or?
It's only a matter of extraction before, because then we filter the supernatant, and we do not have particles. Okay, we are supposed to not to have particles. We only have the reagent.
Thank you, Professor Suleiman and Dr. Lambert for this deep presentation. In fact, I have two questions, one for each. Regarding Dr. Suleimani, the extraction method, you said that you compare, you're using different solvent for biocompound extraction in the Sacra and Teresa, the other type, Serrata.
Yeah.
You use more than one method for extraction and two different type of frankincense. That's Bassola Sacra and Bassola Serrata.
Yeah.
So the difference that you found, is it that significant of difference? Which method is, you said the green method and the other type of method? If you explain just to know which method, which method you found it is more active than the other one.
Yeah. First, we have recuperated the sample of Serrata from colleague in India, which prepared the Serrata by extraction, hydroalcoholic extraction.
Okay.
And we compare this just to know if the extraction by hydroalcoholic solvent impact the final properties of our preparation.
Right.
But we need some
Further
supplement study to compare with the same sacra, with same extract sacra, hydroalcoholic and grandic and green extraction. But this is previous for the next time.
Okay,
We
so
just
your
know,
future.
but for comparative between different fractions, we had released this by quality comparative. We not know in the level of our study, we not compare by statistical or by method of analysis. We just compare between this quantity, but of quality we have not make application of statistical study.
Yeah, because
but we need other more than this.
That's why, because I saw the numbers there, it is almost close to each other. I don't know whether it is significant different or not. So this is, as you said, that it needs more study or more analysis of statistical analysis until you make sure that there is a significant difference of the two methods. Okay, thank you. Regarding the second question for, I think, Dr. Lambert, if possible. After you get the bioactive compounds from your extraction and using this kind of solvent, which is usually known as a toxic solvent, I just want to, did you test the sedimentation or if there is any traces of your solvent in your pure compound to make sure that it is free of any toxicity for, in case you use it for live cells in future?
We have done some tests of compounds only, but it's always with DMSO because we cannot use it in another way to put it on the cells in the cell. And the cells are in water buffer, so we cannot make the comparison of.
So you didn't?
No.
No.
We
So
don't know how we can do it. If you have an
most
idea.
important, you know, because you will use it for human in future or.
Yeah, yes, but
Yeah,
we can
so
only compare with DMSO. We cannot compare without DMSO because we cannot dissolve it.
Yeah. Thank you so much for all both of you.
We just have one slide to comment.
Just this slide for discussion, just 1 minute, to know that why my question is my way to continue for pure compounds from Sacra.
Yeah.
This is question to stop here or to continue for purification of this isomer phosphoric acid. I think that it will not interesting because I believe in activity by synergy between chemical group, between phosphoric acid and other compounds. This is important for green pharmacological properties. And what next time, what do with this work? I present you for functional botanical ingredient. This is not pure compounds. Ingredient is substantial compounds. This mixture of
different
biomolecules. This is important because market of today, 2.8 billion for this market of ingredient in different topics. In the 2030, it will be 500 billion. It's very important to know and to maybe opportunity in the
for future
next time for Dhofar and for Middle East and Africa. This is to elaborate green technology, green process to prepare new ingredient and maybe new biocompound molecule. Pure.
Hope so.
Thank you very much.
I invite Dr. Muhammad Salim Al-Marhoon.
Oh,
good luck.
Welcome.
البوينتر وين؟
هذا للمزيد امام هذا الوراء وهذا الروح
الليزر. السلام عليكم ورحمة الله. هذا معها نفس الشيء ولا؟ شوي هذا أعتقد صوتي عالي وكذا. I am Dr. Muhammad Al-Marhoon, Senior Consultant, Urologist at Sultan Qaboos University. I am also the President of Oman Urology Society and President of Arab Association of Urology. Thank you for the invitation to participate with you our journey in our interest of LUBAN research in the treatment of renal stones. So my topic today is treatment of renal stones with frankincense, a dream to be translated into a clinical trial, because this is our ultimate aim. I'll be going through the introduction, and then I'll take you through the story of our research from animal study, and then our plan for the clinical trial. As you know, renal stone disease, which is urolithiasis, is a common and predisposing factor for urinary tract infections and as well as kidney diseases like chronic kidney disease. And the global incidence of urolithiasis is really increasing. The burden of stone disease is becoming more and more. There is a male predominance, and the incidence and prevalence of urolithiasis depends on the geographical variation, climate, dietary habits, water quality, occupational exposure, inheritance and genetic, latitude, and comorbidities. The high incidence of stone disease in the Gulf region is probably due to the following factors. We have usually a lot of protein diet, so this is one of the factors, especially in Dhofar, I believe. And the environmental factors that we are in a hot, dry climate countries. Is in the stone belt area. It is a country because of the hot, dry weather. We, as inhabitant of this country, we are more prone to formation of stone disease. As you can see here, that other countries in the stone belt, the Arab countries, Egypt, Sudan, Saudi Arabia, United Arab Emirates, and Oman. We have looked at stone prevalence in Oman, and these figures will be as the rate per 10,000 population. So you will find that the outpatient visits of male and females is around 902 out of 10,000 population. And the inpatient admissions for stone problems, whether surgery or admission because of infection, is 74 per 10,000 population. In our study that we have done in 2015, we've looked at the composition of renal stones, and we have found that calcium oxalate constitutes around 44.7%. The calcium phosphate, 3.1, the uric acid, 15.7. But interestingly, the cystine hereditary stone is around 4% of our study population, which is quite high. Because if you look at the international figures, it is less than 1%. So we do have a lot of genetic predisposition for stone disease in Oman. I'm just going to take you through the journey of what we have done so far in this field. As I have mentioned, we have published this paper which looked at the composition of stones in Oman. I have also participated in a book chapter about hereditary kidney stones. And in 2023, we have done the animal study where we looked at the effect of Luban water extract on the treatment of renal stone in rats, which I'm going to touch on. And our next step, which we have not started, but we are fully prepared, is to go for the clinical trial and look at the effect of Luban in renal stones. I have presented part of this animal study last year or 2 years back in this meeting. And just to remind you that we have looked in a rat model of 27 males and 27 females of Wistar Kyoto rats, divided into 9 groups. And we treated the rats with the standard treatment of Urolite U, as well as the Luban treatment at different concentrations. And in fact, in this study, we looked at a treatment as well as prevention. For prevention, after inducing the stone formation by the hydroxyproline, L-proline, for the treatment, we started from the beginning by giving Luban at different concentrations. Whereas for the prevention, whereas for the treatment, we have given from 15 days of the experiment, as you can see here. And then we ended the experiment at 28 days and 29 days. And during this experiment, we have collected biochemical and histological studies. And in fact, we have proven that Luban has shown significant improvement in the treatment and prevention of the experimentally induced renal stones, particularly at the dose of 150 milligram per kg per day. Because we have tested different concentrations, 50, 100, and 150. We do not have any previous studies to touch on which is the best dose, but we have looked at other clinical studies looking at Luban and rats, and we marked our start from this. Definitely, further studies are needed on the effect of Luban to look at another or other animal models. And of course, the next thing is to go to the important part, which is the clinical trial. Now we want to apply whatever basic science we have, whatever studies on animal models, we want to apply it on humans. So this is a collaboration between Sultan Qaboos University and Dhofar University. And the title, as you know, is Treatment of Renal Stones with Frankincense, Phase 1 and Phase 2, randomized placebo-controlled clinical trial to assess the safety and efficacy of novel Boswellia sacchara extract. I'm just going to share with you our study design, and I will be very happy if anybody has input into this, because we have not started, we are just in preparation for this clinical trial. So the idea is that we are going to start Phase 1, Phase 2. That means Phase 1, looking at safety and proper doses, and Phase 2, looking at the effect, the efficacy. So we'll be starting with 34 patients with inclusion and exclusion criteria. The inclusion will be adult, male and females who are having renal stones of 1 centimeter and below, and the exclusion, any patients who is having chronic kidney diseases or cancer, for example. Before the study, we'll be looking at the biochemical, urine, and radiological tests to characterize the basics and look at the stone size, location, if possible, we may infer the composition from the radiological studies. And as I said, in phase 1, we'll be trying the different concentrations at different dosaging just to see what is the best. Of course, you may ask on what basis you have looked at this. Really looking at the literature and seeing any available studies, any animal studies that can give us an idea of proper dosing, as well as we have based our dosing on different toxicity studies. In the phase 2, which is once we have determined what is the proper dose and the frequency of the dose, we'll be randomizing 2 groups of patients, placebo, and which is a control group, and patients. 8 patients, 4 of them will be having calcium oxalate stones, and 4 of them will be having uric acid stones, just to look at different stone types. After that, we'll be following the patients with the biochemical and radiological studies, and we'll follow them at 1, 3, and 6 months. Then we will analyze. Our primary endpoint is stone reduction. If stone size is reduced by 50%, or if stone disappears, this is our primary endpoint, as well as disappearance of symptoms. If the symptoms are disappearing or at least reduced by 50%, this is also a primary endpoint. The secondary endpoints will be toxicity and side effects. Thank you very much. Please.
Thank you for the presentation. I am not a medic, but I have a couple of questions. So would you exclude and exclude the criteria people with recurrent stones? So would this be for patients who have first-time stones, or does it matter?
Because it's a study, we want to exclude any other effects, so definitely we'll be going for first-time stones, and we are limiting to 1 centimeter stone so that we can look at the size and monitor it.
And how are you measuring the toxicity?
Toxicity of the LUBAN. This is based on other studies. And clinically, we'll be, as per protocol, we'll be looking at all parameters of the patient. Symptom-wise, we'll be interacting with the patients. If they have any new feelings, all these will be monitored as per the clinical trial protocol.
Just considering the doses, I see you recommended in the first stage.
Yeah, can we go back?
10,000 milligrams twice daily. I think it's too much. For my experience, when I give for brain tumor, for example, I give 4 grams or 4,000 daily total doses, and somebody developed problem with nausea and diarrhea. Do you think it is not too high? For me, this is extract or not?
We'll be looking at capsules mainly. We are still in the development of the drug.
Is this extract?
No. You are talking about water extract?
No, just Boswellia extract.
It will be the powder which contains the material.
Yeah.
Yes.
Yeah, that is, yeah. But
okay, so regarding the dose, as I said,
I think
we
be
do not have touch base to base on. That's why we have looked at all the literature, and we are trying to see what is the best, most effective dose. This high dose, why we have tested at different levels, because we want to see, let's say at 3,000 or 5,000, we could not find a result. We do not know if a higher dose will be effective. And what will tell us if there are, of course, side effects. Definitely, we are monitoring patients closely, and we'll be stopping at that level. In fact, the water we have used in the animal study, but now we are contemplating using tablets rather than water extract.
خلاص شكرا.
This is clinical trial, humans, yes.
جروب 5 و6 10,000 ملي جرام أي 10 جرام في اليوم من الفوسفيت.
This will be divided. 10,000 will be divided either twice, not repeated. So if I say, for example, 10,000, half of it will be given in the morning, half in the evening. If I say TID, that means that 10,000 will be divided by 3. Part of it in the morning, part of it in the afternoon, part of it in the evening. Because we want to study what is the best dose, what is the best frequency. I will be interested to have any comments. Please share it with Professor Rashan if you are not in touch with me, because he's collaborating with me. And definitely, this is a big concern for us that we want to start in a proper dose that does not push us to side effects. Definitely. Yeah. Please.
الحمد لله صار هنا وهذا الجيم
الباوربوينت عندك؟
هذا ترجع للوراء وهذا الأمام وهذا الليزر.
اوكي. انت اوكسيدنت
واحد يمكن ما نزلنا احنا بعد.
بلا انت اوكسيدنت
هذا لا هذا المرهون. عندك فلاش؟
انا نزلت اول واحد.
دخلنا بيوزر ثاني جديد.
بدل الحاسب ف ما لا مو First of all, thank you very much for the chairman, and thank you very much to Professor Dr. Louay Rashan and the University of Dhafar for the inviting in our lecture quantitative analysis of antioxidant activity in methanolic extracts of newly developed polyphenolic agents. The contents
First, introduction. Second, the aim. Third, types of medicinal plants. Fourth, qualitative and quantitative analysis. And finally, the antioxidant activity. You know, free radical will play an important role for the damage the DNA and leading to the chronic disease, cancer disease, inflammations, aging, and other diseases. The aim of the work, study chemical compounds in the methanol extracts and identify their quantitative and qualitative of 4 plants containing polyphenolic compounds. Introduction, what is the flavonoids? Flavonoids are the typical example or family for polyphenolic compounds, readily recognize the antioxidant activity, anti-inflammatory properties. 4 types of plant containing polyphenolic, like retin, either L-retin or stereochemistry, levoretin, apigenin, and quercetin. Coumarin is well known as antioxidant nowadays, will be the market, found as antioxidant, anti-inflammatory disease, and a dragon fruit for particular fruits, amuna or annuna. And finally, the well-known and commercial one will be ginger. The methodology will deal with the experimental. First of all, the instrument used will be TLC, thin layer chromatography, and high-performance liquid chromatography, HPLC, with the different detector using the TLC plate, and finally, antioxidant activity after separation and extraction of polyphenol compounds. The first one will be a qualitative phytochemical analysis for the dragon to identify different 4 types of compound, tannins, anthocyanin, and flavonoids, and saponin. Content of the selected 4 plants will be using the activate compound in different concentration, levoretins, dextroretins, apigenin, and quercetin. The TLC plate, the TLC thin layer chromatography will be using the separation for the using the RF value, and the HPLC using the retention time in different methods and calculate the concentration and the area of the curve. What is the antioxidants? There are 2 types of antioxidants. First will be natural antioxidants found in the, for example, vitamins, vitamin A, vitamin E, C, and synthetic type 2, synthetic products will be leading to the, for example, chemical compound for hydroxycoumarin. Now, there is a challenge between free radical and antioxidant activity. And this is this table, table 5, found the percentage of the free radical scavenger using vitamin C as a standard. What is the mechanism? The mechanism will be, this is a free radical pi to pi star transition leading to the charge. After that, delocalization forming the final one, the stable one, a product. The calculation for DPP edge using the DVC percent according to the equation above. You can see and calculate the percentage of antioxidant. This is the HPLC for the different compounds. You can find the different concentration and name according to the peak and the area and the peak using standard compounds and separation quantitative compounds for the polyphenolic compounds. In conclusion, simply assay which prospective for plant in methanolic extract are flavonoids, and free qualitative and quantitative analysis are used in the absorbance. The use of free radical or antioxidant, the substance will be used, the kit used 1,1-diphenyl 2-pacryl hydrazide free radical kit using ELISA and known as DPPH method or assay in common method for the antioxidant activity. Now, this is the geography. This is my country, Iraq. Nineveh in the upper part of the map of the Iraq. This is the Babylon, Hammurabi, Mustansiriya, and Samarra city. This is my city, Mosul, Nineveh, in the north of the Iraq. And finally, this is my college, my university, my college of pharmacy in the University of Al-Hadba University at the Mosul, and thank you very much for the all.
Hello, you can take one or two questions. If there are no questions, I would like to thank the speaker once again.
شوف بس مين اللي هيسأل أول وأركب له المايك. بريك بريك كم وقت؟ بريك ربع ساعة وصار ربع ساعة. ينزل سانكيو دي يعني ولا ايه؟
صورت الفيديو كله كلها حقائق. ما عندك ولا مصدر واحد؟
ايوه خلي على الكاميرا اللي من فوق. عزم اللايف مال يوتيوب عزم الساوند.
Okay, the presenter is different.
Okay.
This will go to the presenter. Good afternoon. Ladies and gentlemen, now we will move to our last session of the day. So the first presenter for today's talk, this session talk is Dr. Ahmad Al-Alwi. Dr. Ahmad is Associate Professor in Department of Food Science and Nutrition, College of Agriculture and Marine Sciences, Sultan Qaboos University. He will be presenting on the characterization of protein extraction from Omani seaweeds. Dr. Ahmad, please welcome.
Okay. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. Good afternoon, everybody. I am, as I was introduced, faculty member of the Department of Food Science and Nutrition, but also I am the director of Center of Excellence in Marine Biotechnology. So I have double affiliation. Still, I am still working at the same university, but I have double affiliation, both at the college as well as at the center. So today, inshallah, I am going to talk about a project that we did, my lab, some of my graduate students, Hala Al-Ma'wali did, which is characterization of the protein from the Omani seaweed Hybinia varioides. This plant, we did some work on it back in 2010, and we found some interesting results. So we decided to continue the work to characterize the protein from this plant. Why protein? There is actually a high demand on protein. Why? Because there is a growth in the population, global population, and this growth on population will require food. And the main element of food is protein. So high increase in population will cause high demand on protein. Also, there is rise on the standards, the living standards, which is actually attributed by high consumption of protein. The natural sources of protein is not sufficient to supply all the demands. So there's going to be a crisis. So we have to look for I would say, non-traditional sources of protein, and one of that is seaweeds. Plus also seaweeds offer sustainable source of protein. Why? Because of, first of all, I will talk about it later. I don't want to talk about everything in one slide. Why seaweeds? Seaweeds because seaweeds, not all seaweeds, but there are many seaweeds that have high percentage of protein. As you can see here, some seaweeds have been identified to contain high percentage of protein, around 47%. Seaweeds also have superior productivity compared to some well-known high protein content, such as soybean and alfalfa. So this is taking us to seaweeds. So in terms of productivity, they are high in productivity, and also they are high in protein content. Plus also seaweeds offer sustainability. Sustainability, why? Because there are no arable lands will be needed, no fresh water will be needed, and also no fertilizers will be needed to grow seaweeds. Plus also 90% fewer greenhouse gases versus beef protein will be produced when will be emitted when we produce seaweeds. Plus also, there will be no, as I said, there is no water needed. If we compare that with soybean, we will need around 1,800 liters to produce 1 kilogram of protein from soybean. Plus also, as I said, there will be no need for arable lands. Now, why we have selected this plant, which is Hybinia barriers? We selected this plant, as I have mentioned, we did some work back in 2010, and we found that this plant contains good amount of protein, which is around 17%. So it has a potential to be used as a source of protein. At the top of that, this seaweed contains significant amount of carrageenan. So we have dual benefit of using this plant. It's a source of, it can be used as a source of protein as well as carrageenan. And the global demand or global seaweed market is increasing because of the demand on seaweeds. And there are high figures, as you can see, for this market. This plant, if we search in the literature, will not find much publications on this plant. Therefore, there are some research gaps about this plant, especially in terms of utilization of its ingredients. So this is why those actually were our motives to work on this plant. In order to use any source as a source of protein, first of all, we need to know the characteristics of that protein. Is it a good protein or not a good protein? So from quantity numbers, it has good amount of protein, around 17%. But if this protein is a good protein in order to be utilized or not, this project answered this question. So we decided to look into the quality of the protein in terms of amino acids, what type of amino acids this plant contains, and also the digestibility of this protein. Can be digested or not? Yes, it contains good amount of protein, good amount of essential amino acids, but can be digested or not? Plus also the functional properties, such as solubility, emulsification, water holding capacity, and oil holding capacity. So it can be used as a food additive or not. If the results are okay, this means this protein is a good protein. It can be used as a food, but also can be used as food additives for certain foods. Then we did the experiments. We collected the seaweed from the far, from here. It's a very rich area with seaweeds. Then we dried the seaweeds. We converted to powder. Then we removed the pigments using methanol and acetone. Then the resulting powder was used for protein extraction. And here, there are different types of extractions of protein, many actually. In this project, we used only one type of method for protein extraction, which is using alkaline extraction. Alkaline extraction was used because it is the best method for extracting protein. Theoretically, it's the best method. Why? Because in the alkaline conditions, most of amino acids are charged, so they can be solubilized easily and extracted. Then the supernatant was collected, dialyzed, and freeze-dried, and then we got the protein. From the protein, as you can see, we did all those analysis. I will not go to the details of the technical details of what we have done because I don't think you will be interested on that. So we did water holding capacity, oil holding capacity, solubilization, and we did look into the we did measure the emulsifying properties, forming properties, digestibility, molecular weight and protein profiling, amino acid composition, and FTIR analysis. The results showed that the protein content in 2010, as I have mentioned to you, the percentage was around 17%, 15 to 17%. But this time we got higher amount, which is around 19.5%. Plus also the carbohydrate content was higher and the ash content was lower. The same lab, same plant, but we got different results because of the different years. Actually, it's beyond than the different years. Here we did dialysis. Here we didn't do dialysis. So here what we have, whatever we have here contained besides the nutrients here or the ingredients, also high amount of ash. As you can see here, the ash is around 40%. So removing the ash is very important. And actually it's one of the challenges when we deal with seaweeds. High ash content, high salts content. So it is one of the challenges to have products from seaweeds is controlling and improving separation of the salts. Then the yield. How much protein we were able to extract by using alkaline extraction? It is 6% from the total plant, 6%. So for 19%, this is the without extraction. Now we got only 6%. You may think this is very low amount. This is very low percentage, is 6%. But if you look to the published data from other seaweeds, you will see that the percentage is not that high, actually. So for other plants, for example, Hemithelia elongata, around similar percentage, and others little higher, and the Hibenia japonica is around 13%. This is why it is very challenging to extract most of the proteins from the plant. So why is that? Because there are different types of protein there, and if we use only one method of extraction, then there is a chance of missing other types of proteins. In terms of purity, the purity is very high, around 88%. Other studies showed lower purities, and some studies showed high purities. Okay, how about the functional properties, water holding capacity and oil holding capacity? These are very important properties when we talk about food additives. As you can see, we have the water holding capacity and the oil holding capacity are both high. And it's very comparable with the Hemithelia elongata. Also, both are high. If you check the other plants, other seaweed plants, you will see opposite. For example, here with Hibenia japonica, the water holding capacity is very high, but the oil holding capacity is low. And the same thing with Hibenia caroides, high water holding capacity, but low oil holding capacity. But this plant, our plant, is both are high. Solubility. In order to use any ingredient, that ingredient has to be soluble in the matrix that we are going to use it in. And water, since water is the main or the major solvent, so solubility is very important factor here. As we can see here, the solubility of the extracted protein is high at the neutral and at the alkaline conditions or alkaline pHs, but it is low on the acidic pH. The main explanation for this, because the extraction actually was alkaline extraction. So most of the protein that were extracted actually they are soluble in alkaline, otherwise they will not be there. But the interesting thing here is it's also soluble at the neutral conditions, and most of the food is at neutral pH, not alkaline pH. Emulsifying activity and stability. And emulsifying stability. As you can see, the highest stability and activity are at the alkaline conditions or alkaline pHs. And this is also understood because, as I said at the beginning, the extraction was done at alkaline conditions. Again, also it is stable at neutral conditions. It's not only alkaline pH, but also neutral pH. Also, we have good activity and good stability. Foaming capacity and stability. Foam actually gives some kind of sensation, and it is linked with some foods. For example, cappuccino. Cappuccino without a foam, you cannot call it a cappuccino coffee. So foam in some particular applications is very important. This is why we decided to measure foaming capacity and stability. This graph shows the foaming capacity, and as you can see, the foaming capacity increases as the pH increases, and it's the highest at alkaline conditions, for the same reason that I have mentioned earlier. And here we are measuring the foaming stability. Foaming stability is very important. And if I here give an example about cappuccino, no one drinks cappuccino in the first minute. We enjoy drinking cappuccino, so we enjoy reading, chatting, and while we are doing that, we drink the coffee. So the foam has to stay for some period of time in order to enjoy that. This is called stability. And as you can see here, the stability of the foam is decreasing with the time, which is something understood. And most of the pHs gives high stability, pH 2, 4, 6, 8, and 10, on the first, I would say, 20 minutes. And after that, we see a dramatic decrease on the stability for almost all of them, except one, which is pH 4. You can see that the pH 4, although it starts with low amount, but with the time, it almost stays constant. So in terms of stability, this gives the better stability. For digestibility, we can see that our plant, the digestibility is around 62%. Other plants showed little higher. Some of them showed very high digestibility, and this is actually attributed to a very genuine reason. And this genuine reason is, for this, when we measure this the digestibility, we use the whole plant. And the whole plant contains, besides the protein, some carbohydrates as well as some phenolic compounds. But in these studies, they removed that. Phenolic compounds are known to be inhibitors for the enzymes, the digestive enzymes. So what we try to measure here, we try to measure what is going to happen if we use the whole plant. are we going to get good digestibility or low digestibility? Definitely, if we use pure protein, then the digestibility will be very high. In terms of amino acids profile, the protein from this plant contains around 35% of essential amino acids, which is a good percentage, which means this protein is good in the nutrition value that it gives. However, some amino acids were missing, for example, tryptophan and cysteine. And this is because the method that's being used or that has been used for this analysis actually destroys tryptophan and cysteine. And this is not because of the operator who is doing or the technician who is doing this experiment. No, it's because of the type of experiment. This experiment is known to destroy the tryptophan and the cysteine. In order to measure tryptophan and cysteine, there are other methods to measure them, but we didn't do those experiments. So in conclusion, we can see that this plant, Hybinia barriensis, is a promising source of protein. It's not only protein, but it can be used for other ingredients such as carrageenan. The protein that's found on this plant has very interesting functional properties as well as high quality in terms of types of amino acids as well as the digestibility. And thank you very much. I hope
I would ask you about the 2011 experimental and the study of today. In which month has been taken? Is it in the same month or is it different? And is your study is now, it means after 2018 or before it? Thank you.
Okay. Even if you take from the same place, same month, same day, same hour, the question is not what time. The question is also it's important, but the question is on what growth stage is that plant? Is it young? Is it mature? Or is it old? The composition changes with the growth, with the life cycle. But here what we have mentioned is the percentage or the amount of the salts. This is the main factor there. The amount of salt. This plant contains around 35% salt. 35%. Now, if you remove the salt, directly you are going to increase the other ingredients, because now 35% were removed. The rest will be increased in terms of percentage, and this is what happened here. And as I have mentioned, the main challenge with seaweed work is the control of salt content. How you can remove the salt content from the plant itself.
Because the time we are here in a high gravel. If there is a lot of rain, all of the rain is going to the sea, and the sea will reduce that which is available. If it's available in this area, that means the salinity of the sea will reduce something. It will affect on the planet which is inside. That's why I ask you if it's what time in 2011, the old one, and what time this study, if it's after 2018, means after Mukono. Mukono have been changing a lot of things.
You mean
planet itself.
You mean Gono?
Mukono.
Mm-hmm.
2018.
Gono Muscat.
Yeah. This is all, yeah. Gono is Muscat, yeah. 2008. That's what this is the main
I got your point. But anyways, for all previous studies, all the studies on the plants have shown that the amount of the ash content changes with the life cycle more than changing of the season. And if the changes, if there are any changes, then there will be very little, not much. Yes.
I have time? No. No, okay, no problem.
Can you ask him to take the question?
Okay. Question quickly.
Yeah, that's just, I ask about toxicological effect because
yes.
You have extraction by methanol acetone. I just want to know if you are sure that you have not trace residue of this solvent in your final product. And in the same time, I ask you about contamination. We know that fish as tuna is contaminated by heavy metals like mercury in the water of sea. What about your algae? This is for two questions about safety, because in your bioavailability pH 8 and 10, you know that before to have this pH in a stomachal environment, pH is very low.
Yes.
So, sorry, thank you very much.
Yes. Regarding the toxicity, we haven't done any methods to assess toxicity after the extraction. We haven't done anything. We didn't do any work for that. Okay. Regarding the heavy metals, we did do some work on heavy metals on this plant specifically, and we didn't find any significant concentrations of heavy metals on this plant. Plus also we did on other plants as well, and all taken from the same environment, and we didn't detect any alarming concentrations of heavy metals. Yeah. Thank you.
Thank you, Dr. Ahmad. Our next speaker is Mr.
الميكروفون ولا شو؟
Senior Laboratory Analyst at AST. He will be talking today about anti-mosquito repellent properties of nanoparticle extraction from frankincense leaves. Please welcome.
شوية لا لا عادي ابغى امسك هذا. السلام عليكم ورحمة الله وبركاته. We are here now in the afternoon, so we need to make some energy. I am Ahmad Abdullah Bashir Al-Ubaidun, founder of AST, Advanced Science Technology, and senior chemist at OQBI. I am holding 3 labs, ammonia, methanol, and LPG. Inshallah, today we will talk about one of our products. It's called nanoluban. It is the extraction from the leaves of the luban, and we convert it to the nanoparticle, and we are in the market in the last 2 years. So we will go throughout this story because this is personal issue, then convert it to the research. Now we are in the market. This is all the path. It is starting from the personal perspective. We will go throughout the introduction. What is the main objective of this? The method of the extraction and the conversion of the nanoparticle. And we will go for the toxicity analysis and also the reversal test and the conclusion, then the closure. Everything it is starting from 2022. I am very allergic person against the chemical that uses to control the mosquito. I cannot stay in that place. I have short breathing when I'm controlling to that chemical. So I'm starting from one question. Do we have alternative method to control the mosquito and get against to the chemical? So Before that, we did one survey. We distributed to Salala. 88% from the people also suffering from the same issue. In the GCC country, more than 90%. Then we transfer also for the mosquito, also more than 90%. Also the people is suffering from the chemical. And we know that year by year, the mosquito have ability to modify the target compound that is available in their study or their body to fight the chemical that we are treated. So we need to think alternative. What is the alternative method we can use to control it? Then why we go for the leaves? Even the essential oil, it is important to control the mosquito, but why the leaves? Because the leaves, they have compound, it is not available in the jam or the final product. I need to control the mosquito. I'm not working to the anti-inflammation or another compound. I need to control the mosquito. The mosquitoes, they don't like to work with the like the aromatic compound. So the high concentration of the leaf, the pinene and also the terpene, it is available in the leaves, not in the jam. In the jam, they have essential oil. A lot of the organic compounds is available there. We are trying to control it with the essential oil, but the efficiency is the same like the chemical. So we are transferred to the leaves. So the compound it is the leaf, it is not available in the jam itself. This is why the high concentration of the bean and also the terpene, it is available in the leaves, but it is not available in the jam itself. So this is why we are working to the leaves. The main object of this to extract the bioactive compound that is available in the leaves and then convert it to the nanosize. And then we did the synthesis at the final product. So we are starting from the extraction. We use the simple water as the solvent. We are not use any chemical. Like we are trying more than 10 solvent, methanol, ethanol, the ratio between all those chemicals. The best one, it is the water. The simple to extract, simple to purify the final product as compared to the organic one. Because the organic compound, we will have a lot of the chemical that is available there. So I need to simplify the amount of the purification cost that is available after the extraction. So the water, it is the best one. Then we are shifted to the nanosize. In that stage, we have two options. We can go for the physical one, we can use the microwave, or the microwave technology, we can convert it to the nanosize, or we can go for the chemical one. We are using the chemical one, it is solvent, or silver nitrate, with the concentration 1 millimolar. We take the ratio between the extract and the silver nitrate 9:1, and then we convert it to the nanosize. And this is the main extract on the left, it is the water. And one of the indication that our product it is converted to the nanosize is the color. So the brown color that it is confirmation, we are convert our product to the nanosize. Then we did some experiment in Nazwa University. We have the FTIR, and from the FTIR we have OH group, hydroxide group, and then we have the carbonyl group, as well as the C-double bond compounds that's available in our extract. And then we did the UV extraction or the UV analysis to see we are formation of our silver nanoparticle or not. And the silver nanoparticle have the wavelength between 400 to the 500 nanometer. So we are successfully synthesized the silver nanoparticle from the leaves itself. Then what is the composition in the leaves? Why we are using the leaf? Because the high amount of the terpene and their derivative, limonene and derivative, it is available in the leaves. It is not available in the jam itself. And as I mentioned, I'm focusing to the solvent we are using the water. The organic compounds when we are using, they give us different result. But still the most important thing for us, we are using the water here. So this is all the compounds that is available there. So I'm sorry, I will not give you the official analysis because still I'm not received it from that lab. So this is the approximate what we have it in our extract. This is the psychotoxicity in Nazwa University. This analysis is did against the shrimp in last year, and we give us the LC50 600 ppm. So we are have ability also in the psychotoxicity because our product it is only for 1%. In the market, only in 1% that we are more than or far away from the toxicity level. And this is psychotoxicity. We have the oral actual toxicity. If you are interested to see the full report, you can scan the barcode. And from this analysis, we got our final product, it is free from any oral toxicity. Even you are drinking our final product, you will not get anything. We are not classify even our product is toxic, even for the oral also. This is for the dermal also in the same lab. We are not classified under any dermal actual toxicity. Our product is safe to use in our daily life. And this is anti-mosquito. Successfully, our product have ability to control more than 90% of the mosquito. Even from the adult, from the baby of that mosquito, all level of the mosquito, we have ability to control it more than 90% from the leaves. This is why many people, why you ask me, why the leaves? Because this result. This is the field experiment. In 2023, we did field experiment with Ministry of Agriculture and Ministry of Dhafar. And we see we have more than 5 locations, Sa'adas, Hanot, Taqa, Marbat, and Hariz. We choose those areas because they are suffering from the mosquito. And we see the efficiency of our product. It is 95, 90, 90, 86, 75, and 80. In the last year, we did the same experiment. We see the efficiency of the product even more. So the sustainability of our product is available. And in the conclusion, our product, it is free from any toxicity, as the analysis say. I'm not saying anything. The analysis say. And the water is the best extraction, as well as the most bioactive compound is available in our product. It is not available in the jam itself, as well as I mentioned, the water is the best extraction for that. The last update in this research, we have 2 updates. We are using now, we are synthesize new material, it's called quantum dot. The quantum dot, it is a semiconductor. We are synthesized from our leaves. This quantum dot, I will share this information the first time. We need to protect it, bro. Huh? This quantum dot have ability to detect the bacteria as well as the mosquito in our environment. That means when I am spray our product in the area, I need to see how much the mosquito available in that area. So our quantum dot is synthesized from this, have ability to visualize this. We are using the drone to see how much or what is the population of the mosquito that is available in that area. From where? From the leaves. We are synthesized the quantum dot from the leaves. It's called carbon quantum dot. We are synthesized it from there, and we get very excellent result after this synthesis. As well as we are now working with same quantum dot to detect the ctDNA, the circulated tumor DNA. It is available in our blood to check the early stage of the cancer. It is available in our blood. Everything it is in the leaf. I will repeat it again. This is why we need to concentrate it. Sometimes maybe the leaves will have solution. It is not available in the jam itself. So this is the result for the 2 years. And still we are going on to discover what is available in the leaves. I need to acknowledge definitely in Nazwa University, OQBI lab, as well as the SIG lab, as well as myself. Because I am working to a lot of things. It is happening in the last 2 years, and the research team. This is from my side. I think I'm not taking too much time.
Thank you so much for insightful talk. We will have one or two questions.
Very interesting subject. However, regarding chemical analysis of Luban leaves, you mentioned that you managed to extract boswellic acid. Which one, please? Which boswellic acids?
I'm not trying to extract the boswellic acid. I need to extract the billitine and the trypene. It is available in that leaf. Also, I am now in the stage I need to configure which one to have or the bioactive compound against the mosquito.
Yeah.
Still, I don't have that information. I have the mix. It is available in the leaves. I make our analysis to that. Now I am in the purification stage. I need to purify, so I will test each fraction it is available in our product.
Because as far as I know, leaves, they don't contain boswellic acids.
Very low concentration.
Even low. Even low. We used HPLC MS, which is more sensitive than the method you used. We didn't find any boswellic acid in the leaves, as well as in the root, as well as in the fruit.
Yeah.
But we found boswellic acid in the bark.
Okay.
In addition to the gum resin.
Yeah, this is why from the beginning I told you I'm not interested in the boswellic acid.
Yeah.
This is why. I know maybe we will find it in the low concentration. This is why from the beginning I don't consider the boswellic acid. Because I know maybe if I will find it in the small concentration or not.
All the best. Thank you.
Any other questions?
I want to ask one question.
Yeah, tell me.
Is it works as a repellent or is it work as a repellent or killing or lethal to the mosquitoes?
It is working as a repellent with the adults.
With the adults?
Adult mosquito. But for the baby or for the age or the first stage of the mosquito, they have ability to kill.
Thank you.
Any question there? Yes. Yeah.
And you have used this leaves extract to prepare silver nanoparticles, 9:1 ratio. So the activity you are reporting, that's from silver nanoparticles? I think so.
Yeah.
So then how come it is for nanoluban?
Yeah, nanoluban, that means that we use the extract at the reduction of the silver nanoparticle.
Yes.
So we are, this is let's say it is the commercial name. See the nanoluban, that means commercial name.
Is not luban but silver nanoparticle.
Yeah, silver nanoparticle, but if we are talking about the commercial one, it is nanoparticle. Nanoparticle from the where?
Means all the activity you are claiming is due to silver nanoparticle?
No. Mostly we are focusing in the silver nanoparticle and also the extraction from the leaf itself.
What I mean to ask that, what's the concentration of nanoluban then?
See, the nanoluban, it is, we are working to 9:1 concentration. So the concentration of the nanoluban is available there, 50% as compared to the silver nanoparticle, it is 50%. I will explain it to you. I will come to you, I will explain.
Thank you so much.
Yeah.
Just know your product. When we, against fly insect, we use insecticide or repulsicide.
Yeah. Yeah.
Your product is insecticide or repulsicide?
Repelliticide. Yeah. Okay. It's not insecticide.
But repulsicide, I am very surprised because in repulsicide is not induce deed of insect. It's not toxic.
Yeah.
But you have said that you show that is toxic for against fly, insect, mosquito.
This is the most interesting. The most interesting part in this.
Repulsicide. Okay.
Thank you.
Thank you so much, Mr. Emad. So our next speaker is Professor Sajid Jogestu. He is professor in Department of Marine Science and Fisheries, Sultan Qaboos University, and chairholder of Industry Chair in Marine Biotechnology. Today, Professor Sajid will be talking about the microbial and fungal activities of standardized Omani techniques.
Okay. Good afternoon, everyone. I'm also, as Dr. Ahmed Al-Alawi, affiliated with the Center of Marine Biotechnology at Sultan Qaboos University. And today I'm going to talk about frankincense, in particular standardized powder, extraction powder from frankincense. This is outline of my presentation, so it's quite standard. I will not go through this. This is an introduction, and most of you already know a lot about frankincense. We are quite new in this field, but frankincense is an aromatic resin gum that can be obtained from Boswellia trees, and in Oman we have Boswellia sacra. So we are working with this species. And the main component of Boswellia includes different boswellic acids, alpha and beta boswellic acids. And the composition is varying. We heard that the different species of Boswellia, so we have different extracts from these different species, even the same species obtained from different seasons and different environment. They might also have a variability in extracts and composition, and that's create a lot of problems. So standardization and having particular composition of the gum resin is particularly important. This is the slide that I demonstrated 2 years ago on the previous meeting. So we were interested in the antimicrobial activity of essential oils comes from frankincense, and it's demonstrated basically when we mix it with the chitosan. So we have 1%, 3% chitosan mixed with the frankincense oil, essential oil. It's increased antimicrobial activity. So it's showing a number of bacteria as colony-forming units. Beside of this, frankincense has antifungal activity, and we can prevent growth of numerous yeasts and molds. But again, activity varies with different species of bacteria or fungi are used, as well as extraction methods. So again, there are a lot of variability in this regard. How this standardized frankincense powder was obtained. Again, Professor Lashan provided us with this. Again, he can maybe comment if I'm wrong in this. So basically we have acid-based extraction with ethanol water, and then filtering, washing, and then drying step under vacuum at 75 degrees. The objective of this study was to test the antimicrobial properties in terms of fungi and pathogenic bacteria of this frankincense powder obtained by this acid extraction method. For the results, my project had 3 parts. First, analysis of the composition of the extract. Second, testing activity against gram-positive and gram-negative pathogens such as E. coli and Staphylococcus aureus, and testing against fungal pathogens, plant pathogens such as Fusarium and Alternaria alternata. For the analysis of the composition, it was done by help of Professor Rashan. So we used we dissolved the sample in methanol, and then we use LCMS MS with the C18 column, where we use solvent methanol and acetic acid with the flow of 0.6 milliliter per minute. And then we also use the standards to see if these compounds are present or not. For antimicrobial activity, we used 2 bacterial species and 2 fungal species. Basically, we dissolve extract in methanol, and then we applied at different concentrations to sterile discs. Then methanol was evaporated, and then we incubate these petri dishes for 24 hours in terms of bacteria at 37 degrees, or 5 days in terms of fungi. And then at the end, we measure inhibition zones. So the results are coming here. This table shows the composition of the extract. So the major component was beta basphalic acid, so acetyl beta basphalic acid, so about 86 microgram per milligram of the extract. And less dominant were beta basphalic acid, and at low amount we have lipoleic acids such as alpha lipoic acid. So you can see that this is ALA was at 42.5 microgram. This is the results of antibacterial activity. So again, we had Escherichia coli and Staphylococcus aureus, and you can see that results were quite surprising for us. It was quite weak, or we can say moderate activity with the minimal inhibitory concentration, about 500 microgram per milliliter in case of E. coli, and the Staphylococcus was even much higher concentrations. However, in terms of antifungal activity, we can see that we had strong antifungal activity of the extracts, especially in case of Fusarium, and the growth was inhibited at the concentration of 0.25 microgram per milliliter. In case of Alternaria, we saw some effect at the concentration twice higher, 0.5 milligram per milliliter. These slides, we try to investigate the possible antimicrobial mechanisms. Again, this is the next step that we want to approach now to understand how this, especially antifungal activity, happened. We believe that it could be due to different reasons, possibly induced oxidative stress by generation of reactive oxygen species, inhibition of enzymes, disruption of cells, as well as it's known that some of the compounds from frankincense can reduce fungal spores germination and hyphal elongation. So in conclusion, we can show that the main component of that standardized acid extract is acetyl beta basphalic acid. There was a low antibacterial activity of that extract. However, it was high antifungal activity against plant pathogens. And it is possible to use these extract standardized effects for different industrial applications, in particular, probably to prevent growth of plant pathogens. Thank you very much. And a special thanks for organizers, Professor Rashan, and my postdoc that work on this project, Dr. Laila Namani. Thank you.
Do you have any idea or plans to investigate the effects on, or the effect of boswellic acid on penetrating biofilms? Because what you've shown is really robust data on the antimicrobial effect, but I'm assuming it's a single species, not a complete biofilm.
Mm-hmm. Thank you for your question. It's actually a good question because 2 years ago I was presenting the talk regarding using chitosan and extracts from frankincense, and that was a work done by my master student, and she had the two projects. One was on biofilms, marine biofilms in particular, and another one on extension of the shelf life of strawberries. So we found out that frankincense, at least for marine bacteria, is not working well, but antifungal activity for plant pathogens, it was really remarkable. I hope I answered your question.
Thank you so much for the insightful talk. We will move to our next speaker now.
Can you remove?
Mr. Said Mustafa Batik Farouk. He is engineer at the farms.
حطيتها فوق. أحسنت. It is there. It's better to. السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف خلق الله سيدنا محمد وعلى آله وصحبه وسلم تسليما كثيرا ما بعد. Welcome to our study which is about the medicinal and nutritional potential of Acacia Senegal, as it's called gum Arabic tree in Dhofar, Sultanate of Oman. As you know, that gum Arabic tree is a tree that belongs to the Phoebus family with a cylindrical flaky trunk which is reaching up to 6 meters, as it is appear in the height. And also the scientific name of this tree is Acacia Senegal, as it's common, but now there is a reclassified that they call it as the Senegalia Senegal. In the region Arab country, they call it as Hashab in some area, and also some area they call it gum Arabic tree. In Dhofar, they call it as Thur in different area, and Thummar, and also Thumur in Mahri language. First of all, we'll talk about the global distribution of this tree. As we can see from the map, it is on the African Sahelian belt, which is from the Senegal in the west into the Somalia in the east, and it's also goes to the Arabian Peninsula and also reach India and Pakistan. In the Arabian Peninsula, just only found it through the study only in Dhofar region. So it is unique here, only in this area. On the past study, we found some memories on the bridge console of a bridge officer who speak about the Arabian gum trees. For example, Heinz, he is a bridge officer. He said that Dhofar coast was the main coast for the gum Arabic production and also frankincense. And also Hunter in 1877, he said that the main product that export from Aden was gum Arabic and also frankincense. Thomas, he's a British traveler. He also noticed that, observed that there is a high number of those trees in the valley and on the plains, and is excluded naturally from the branch without human intervention. There is some recent study, Miller and Morris, they write a book about plant of Dhofar, describe the gum Arabic tree, and also Al-Kthiri, Ahmed Samdid Al-Kthiri, he has two study. One study highlight the tree geographical distribution, and the other one which is discuss the importance and gum production season. Also, one guy, Al-Sheikh, in 2013, he make a study on the vegetation cover on the Wadi Garziz. Another study compare between the gum Arabic from Sudan and the Dhofar region, which is Al-Alwi in 2018. So the issue is there is a scarcity of previous study on Omani gum Arabic trees, and also there is a confusion on the naming, either on the plant itself and on the gum also. So we highlight the importance of Jama Arabic tree, and also we emphasize the medical and economic value. Also, we clarify the convention on the tree. The study was done by field visits and interview with the 10 elderly narrators represent different environment of Dhofar. Also, we cross verification of information through the reviewing both ancient and modern source and compare them with what we get from the people. As you know that, the acacia genus is a high genus that consists of about 1,380 species. So, and there is high similarity between some of those species. For example, like Samar, Qarad, and Talah, and Qatad. Those variety is overlapping with the names with the Samq al-Arabi, with the gum Arabic trees. So we can notice from the study in Arabian lexicons, we found, for example, in Al-Razi, Ibn al-Mandur, Al-Zabidi, they call the gum Arabic tree is the Talah. In Al-Muzaffar dictionary, he said that Qarad. In Wellsted also, he's a British officer. In 1838, he said that Qaf and Samar are the gum Arabic tree. Hunter also said that Arabic gum is extracted from Talah, Samar, and Qarad. So we found there is many different types of gum Arabic tree. FAO, Food and Agriculture Organization, they, in order to unify the names, they say that the only gum Arabic tree is that come from Hashab, which is that what we have it here. And recently they add Talah, which is the Acacia Ciala. Another overlapping in the name of the gum or resins. Here in Dhofar, we call the gum itself, we call it as mloch. This mloch is named also with other plants. That the gum that come from Saqot, the gum that come from Talah, the gum that come from Kthora. Those all are called it as from 6 trees. So there is also overlapping on the names. Now we will move to the distribution of this tree on Dhofar. We can see on the mountains, it's along the Dhofar mountains, especially on the upper part of Garbeb, which is the plain, and also on the slopes edge that interface the wind that come from the sea. Al-Kthiri, he said in his study, the average density of the Thur tree in seasonal forest is around 81 tree per hectare. Also, a sheikh, he said that when he did his study on Wadi Jarziz, he said that around 210 tree per hectare. So that means there is a high number of tree on that area. Moving to the gum Arabic production, the name of gum Arabic is a natural viscous exudate that appears on the tree trunk due to tapping or injury caused by natural or human factor. Like what? Oops. Yeah, like what we can see here on the tree. And there is two types. The composition of this gum consists of carbohydrate and dietary fibers and contains various nutrients. There are two types in Dhofar region. One type is called crystal white, which is we name as Jehozah Thur. It is white one like this, and this is useless. The other one dark red to brown, which is in this photo. This is the common and commercially used type. The collection season for this kind of, for the gum arabic is during February and March, after winter season. And the tree yield, according to study, it ranks from 20 grams to 2 kilo per tree, with an average of 250 grams per tree. And the natural factor that affecting the gamma with the tapping is the strong north wind during winter season, high humidity and high temperature, also the seasonal rains. There is a medicinal importance of this tree. For whole tree, we can notice that the bark, it's used to treat the wounds and clean them from the infection and gangrene. Also, the camel milk that feed from this tree is considered as the highest quality type of milk with excellent nutritional benefits. Also, the gum itself, it's traditionally used in Dhofar to treat chest and lung diseases, digestive cleaning, and back and joint pain relief, stop bleeding, and also for the treating the stomach ulcers, and also for the diabetes and high blood pressure management, and also for boosting the immune system, also used for bone fracture stabilization. But nowadays, they only use only with the elderly people. There is a potential use on the gum arabic in the world, as they call it safe food additives. Since 1998, they recorded, they approved as a safe food additives, and they include in many products such as the soft drinks. Also, it's introduced in the medical application, and they use it on the capsules and tablets and medical bufferings. Also, they use it in the cosmetics. Also, because of the extremely low in calories, which is less than 1 kilo calories per gram, so they make it in the dietary program, or also weight loss and cholesterol reduction. The global quality, the highest quality for this Arabic gamma is from Sudanese, but according to a study that we go through it, which is Al-Alawi, he demonstrated that the Omani gum that's come from Dhofar is as effective as Sudanese gum in treating the diseases as a source of natural antibiotics. Here are some recent statistics on the gum arabic production and trade, and we found around 168 thousand tons. It is in the global production, and Sudan has the highest share with 66%. In Dhofar, the current gum arabic production is very scarce and nearly discounted, and the local demand is minimal, mostly limited to the elderly consumer, as we said. And the traders avoid to market this because of fluctuation production and also poor financial returns. And this is the price of, the different price for the ton of gum arabic international market. This tree also, the uses of other parts rather than the gum itself. The bark also used for the leather tanning. Also the thin stems and branches is made into the fish traps. Hardwood also utilized in the construction. The leaf and flower, highly nutritional fodder for the animals. Also the fruit, the buds, preferred part for the animals, rich in nutrients. And also the roots used to fix the nitrogen in the soil. So it's improving the soil fertility and agricultural productivity. One of the good products that the honey producer use this tree to produce the gum arabic honey because of the dense spread of the gum arabic tree on the Dhofar. As we can see here, the thorn tree, which is the gum arabic tree with the fully of flowers. During the month of mid of March to mid of April, the flower season. So the honey producer use those trees in order to produce the dabish al-thorn. They call it in locally dabish al-thorn, and it is one of the unique honey. And we can see it is yellowish and very rare in the area and the region. And the production, we estimate the production according to what we meet the people. In 2022, it's reached not less than 3 tons. And also the price range from 20 to 35 Omani Riyal per kilo, with an average 25. So the thorn tree is founded in the Dhofar Mountains, is same as the gum Arabic tree, which is unique in Sultanate of Oman. The wide distribution of the tree in the Dhofar Mountains today, along it is continuous gum Arabic production, confirms what we found on the literature on the past, the figure that reported by the previous in 19th century studies. Also, the production of Omani gum Arabic continues, but only in small and limited quantity. However, the scientific studies confirm it is quality. And also, the local gum Arabic production occurs naturally without human intervention, and it is influenced by the natural factor. Nobody use it like what it in Sudan. The human interact with it. And also, finally, the gum Arabic honey is produced in Dhofar in large quantity and high quality, enabling it to reach a regional and international market. Finally, we recommend to conduct qualitative research on the gum Arabic tree in Dhofar and carry out field survey to accurately map its distribution and also to maximize the utilization of gum Arabic to make it a significant contributor to the national economic. We would like to thank the researcher and narrators who were interviewed or contacted for providing us valuable information. And also we extend thanks to the organizer for this meeting. Thank you so much.
Now I think this, as he suggested, more research is required on this, and other researchers related to this field are here. So is there any question? Or you can go ahead and share with anyone. So one question.
Thank you, Engineer Said. Thank you about your presentation. I want to know the production of the gum of thorn. Is come naturally, or it need to the human to reduce or make some crack on the stem of this tree? And you said it's very low production per tree. I think comparing to the same species in Sudan. What's the reason of that? If you have any idea about that. Thank you.
Thank you for the question. In the production here, they just occurs naturally without any cracking, but in other area, for example, in Africa, they invest in this area. Even they make like what we make in Luban, typing. What we make for Luban, they make also for the Acacia Senegal. So here we didn't we keep it as natural. So we can invest on that also here.
Thank you so much.
Thank you.
We'll move to our next speaker.
I have small information to add. Thanks for the presentation.
Thank you so much.
I just want to add, when you are mentioning about the uses, this species is also used as a dye extraction.
Yeah.
In India, we are using this Acacia Senegal for yellow color dye from the leaves.
Dyeing, yeah.
Yeah, so that is additional value. In fact, apart from the gum, maybe this plant can be explored for yellow color dye, which can be used for textiles as well as for cosmetic industry.
Yeah, of course. Thank you so much.
So final speaker for the session is Professor
Allah يعافيك يا رب. الله عليك
University College, Iraq. Today he will be discussing about biological evaluation of new curcumin derivatives. Please welcome Professor Ahmad.
We have this slides.
بسم الله الرحمن الرحيم والصلاة والسلام على رسول الله محمد وعلى آله وصحبه وسلم. I would like to express my sincere thanks to Al-Dhafar University and my truly thanks and appreciation to the organizer committee of this third international conference or meeting on Frankenstein or frankincense and medical plants. I'm going to talk about the synthesis and biological activity or evaluation of some new coumarin and or derivatives. First of all, I would like to talk about some notes on what we did before. I am Dr. Badi Alim Ahmed from Al-Hadba University. My specialist is organic chemist. I am not a biologist. I am not a pharmacist. I am an organic chemistry system. Right. We and my colleagues are very interested in doing some alteration in the substituents on the medicinal drugs. We are take all the precautions about the activity and about all the physical properties of molecules. For instance, we have some projects, hundreds of projects we did. These projects concerned with synthesis and approaches to the synthesis of adriamycin, donomycin, and oxorubicin as anti-cancer drugs. We did a lot of work on it and do some modification on the substituents. Move over to the synthesis of nitroimidazole as flagyl, as a trade name. We did also a lot of work on synthesis and modification on the substituents of the groups on the molecule. We did a change in the functional groups from carboxylic acid to amide to ester to anhydride to acyl chloride and then we have a precaution about the stereochemistry of the molecule. We also moved to synthesized captopril or capotin as a heart disease modification. Then we moved also on succeeded in preparation and synthesis of dioxin and another molecules as bile acids, steroidal compounds. We did a lot of work on it, and we managed to convert it to the vitamin D by irradiation. Me and my groups can alter any substituent on any compound, organic compounds, and have modification on the substituents in order to also have stereochemistry and all the stereo part related to this organic compounds. Finally, we move again to our lecture, synthesis and biological activity or evaluation of coumarin derivatives. Will you please take this one? Mm. Yes. Oh, thank you. This compound, this is the new novel organic products. This was characterized by all the spectroscopic and chemical means. This formed by the SN2 reaction or addition of this first moiety and then condensed with the coumarin or aminocoumarin. This aminocoumarin can be synthesized first from any amino group, can attack this oxygen of coumarin and have a new product. Thiazole and coumarin are well known to have a broad spectrum of activity, antimicrobial, antibacterial, antifungal, and so on. This compound have been synthesized easily by condensation of this 2-amino thiazole with coumarin using dry pyridine and to have this new novel product. This part can do a lot of things. The first is as antimicrobial, this is an antibacterial, and this is or the thiazole itself can act as antibacterial, antimicrobial, antioxidant, anti-tumor, and so on. Then the condensation of these two parts can give us an antibacterial significant effect on this is an antimicrobial as a whole, and this is the table for showing us the functionality of this new product. Again, this is an antimicrobial or antifungal effects of some of our substituents. So the synthesis of this murakkab is dead easy, can afforded by two procedures: synthesis of the aminothiazole and then SN2 reaction with the coumarin. So we got the product and we can use it as medicinal product for any as antioxidant, anti-tumor as well, and antibacterial. This is the production of one of our products. We have more than hundreds of papers related to the medicine or medicinal products. Conclusion, of course, those products, the quinolone, this one, quinolone, is derived from quinoline as the first, and can be used as anti-cancer or anti-tumor drugs. Then when we condense both of them, we have a high significant activity against the on scanning this compound on the antibacterial and antioxidant and anti-tumor. We got a very high significant results. Anyway, those products compounded from the thiazole, 1B, C, E, and G as substituents can have a very high efficiency in terms of antifungal. Also, the second compound, the product, final product, also can have this activity. Thank you for your attention, and if there is any question, we can answer. Thank you.
We can take one last question.
Thank you. Thanks a lot.
Okay, so if there is no question, then we can conclude the session for today. Tomorrow there is we're going to go someplace session.
يا الله. الله الله يبارك فيك. أحسنت.